Everyone, my name is Sujata from BSc final year and my teammates are Kaushik, Aravind, Ankita, Madhu and Sandhya from BSc second year. We are here to present about Jignasa student study project on the topic of Piper longum from Department of Biotechnology. So out of curiosity, we have done some experiment to know about the Piper longum and Pipalli Magada Krishna Vaidehi Chapalakarna Upakulosana Shaudi Kola Satya Krishna Tandula. The meaning of the sentence is Pipali can be used as an antioxidant properties and also cure many diseases like fever and all. And today objectives of our project is to find the nature of phytochemicals present in the Piper longum and to evaluate the antibacterial activities of the Piper longum. So, Pipali is a most popular spice in India. It was used by the Greek, Rome, and Europe also for the preparation of the medicines and for preparation of the wine. And its medicinal properties and nutritional properties was described in the book of Ayurveda. And the Pipali was grown in evergreen forests, mostly cultivated in the southern part of India. Pipali has many phytochemical compounds, and present research also said that Pipali has anti-cancer and antioxidant properties.
The foods are used as spices and also in pickles, preservatives, foods. The most important use of pepper longum is medicinal ingredient in Indian medicine system, Ayurveda, Siddha and Unani. Indian long pepper is used to improve appetite and digestion. Pipali may be used in managing digestive problems. Research and methods. Collection and extraction of plant material. Dry fruits of pepper longum were collected from local market Hyderabad. Screening test for phytochemical constituents. Qualitative test for samples, tannins and alkaloids. Test for tannins. 0.5 grams of dyed powder was sample boiled in 20 ml of water. 0.1% ferric chloride added and observed bluish brown colors. Test for alkaloids. 0.5 grams dyed powder sample was boiled in 20 ml of water and filtered. Add Mayer's reagent to see creamy and wet precipitate to indicate positive test. Test for samples. 2 grams of powder sample was boiled in 20 ml of distilled water. Finally, the froth was added in with 3 drops of olive oil, shaken vigorously and observed the formation of emulsion. Phytochemical result for phytochemical tests. Samples positive, tannins positive, alkaloids positive.
Evaluation of antimicrobial activity. Preparation of plant extraction. The 6 samples were prepared with using Piper longum powder. The preparation of first 3 samples is similar to another 3 samples. But the difference is their incubation time. Means first 3 samples were incubated for 5 days. Another 3 samples were freshly prepared. In those add 2.5 grams of Piper longum and dissolve them in 30 ml of distilled water, ethyl acetate and glycolic acid respectively. Sample 7 and 8 contains 50 mg per ml of amoxicillin and azithromycin respectively. Microbial cultures for antibiotic assay. Gram-negative bacteria and gram-positive bacteria were chosen for this assay. Gram-positive bacteria such as Actinomycetes, Lactobacillus and Bacillus and gram-negative bacteria E. coli. We have collected Actinomycetes from random scraping material at wash area and dissolved it in 10 ml of distilled water. 1 ml of curd and 1 ml of idli batter were the sources of Bacillus 1 and 2 species. 1 ml of LB broth contains DH5 alpha cells were the sources for E. coli cells.
Cereal dilution and obtaining pure cultures. 1 ml of source material was dissolved in 9 ml of sterile water. For each sample, 10-fold dilution was made. Subsequently, the same method followed. 4 cereal dilution samples were plated on freshly made LB agar plates. Later, these plates incubated at 37 degrees for 24 hours in incubator. Well-separated single colonies of each sample were picked up from the LB agar plates and incubated in LB broth. The culture obtained in LB broth after overnight incubation were microscopically evaluated and treated as pure cultures. Obtained pure cultures were later used for disc diffusion assay experiments. In disc diffusion assay procedure, liquid culture were obtained by diluting 1 ml of pure culture in 9 ml of sterile distilled water. 100 microliter of culture were plated on LB agar plates using spread plate technique. Sterile filter disc dipped in different label solution of pepper longum extract were placed equidistant from the center and labeled. The plates were incubated and later observed for the formation of clear zones. Measurement of zone of inhibition through disc diffusion assay. After incubation, the diameter of zone of complete inhibition starting from the center of the disc. The measurement is done in the sterile environment. The area of zone of inhibition caused due to the extract against standard reference was treated as qualitative MIC or disc diffusion assay.
Results and discussions. Interesting results were obtained with the qualitative disc diffusion assay. Distinct clear zones were obtained in all the cultures. The standard control plates with no inhibitory agent was considered as positive controls, and the synthetic drugs were labeled as negative controls. They are sample 7 and sample 8. And here are the some of the pictures of stick plate method and pour plate method and measuring of the clearance zones. Next, in this figure 4, the Bacillus 1 species, these are the clear zones obtained in the Bacillus 1 species. Here, from this figure, we can say that sample 3 and sample 6, which are the natural extracts of Piper longum dissolved in glacial acetic acid, has shown its inhibition activity more when compared to the other samples. Whereas in Bacillus 2, the sample 3 and sample 6 and also the synthetic drug sample 7 and sample 8 has also shown their inhibition activity. Whereas in Actinomycetes species, the sample 6, which is a natural extract of Piper longum and glacial acetic acid, has shown more inhibition activity than any other samples. Whereas in E. coli species, along with the sample 3, the sample 7 and sample 8, which are the synthetic drugs, has also shown their inhibition activity. And from this report, we can say that sample 3 and sample 6, which are the natural extract of Piper longum dissolved in glacial acetic acid, has shown more inhibition activity and they are comparable to the activity of the synthetic drugs. And here are the samples which I have taken. Sample 1, 2, 3 and sample 4, 5, 6 are kind of similar samples, whereas sample 1, 2, 3 are incubated sample for 5 days, whereas sample 4, 5, 6 are freshly prepared samples, whereas sample 7 and 8 are synthetic samples. The natural samples were taken in the concentration of 80 milligrams per ml, whereas synthetic samples amoxicillin and azithromycin were taken in 50 milligrams per ml. Next, conclusions. From the table 1, we can say that Piper longum has shown the bactericidal activity against all the samples which we have taken. The area of clearance or area of inhibitions are variable, whereas Piper longum obtained using the natural extracts by dissolving Piper longum in the glacial acetic acid in sample 3 and sample 6 has given maximum clearance zones, and the clearance zones which are obtained with sample 3 and sample 6 are comparable to the synthetic standards. The freshly prepared Piper longum in acetic acid sample 6 has shown maximum activity against the Actinomycetes and Lactobacillus species, whereas Piper longum extract in acetic acid incubated for 5 days sample 3 has shown maximum activity against the Bacillus species from Idli batter and E. coli species. And from the above experiment, it is clear that potential of natural ingredients depends varying on the processing, and reasons for the variable potency is due to the altered chemical constituents like dispersion of active ingredient, chelation and etc. This also asserts the fact that our ancestors or medical system have given insights into such indices. Their prescription for same ingredient or drug was recommended in different forms after diverse processing. Such optimal doses of active ingredient reaches the patient. Due to the diverse ethnomedical and Ayurvedic pharmaceutical applications, the demand of people is rapidly increasing in the worldwide and international markets. With the proper cultivation and technology, our country can aim to raise the major global market of the long pepper. And Piper longum is one of the best crops to be cultivated in additional income source along with the various crops like silviculture and horticultural importance. It may also promote the self-employment opportunity to the farmers through processing of the marketing of the products.